So this is part 2 in the chapter 12 lecture on the nervous system, talking specifically about electrical signals within neurons. So, electrical signals within neurons. This is really important because this is basically how information is transmitted through those neurons. So electrically excitable cells are going to communicate with each other via either an action potential or a graded potential. All right. So a graded potential is going to allow for communication over a short distance, whereas an action potential is going to allow for communication to occur over a short and a long distance. So just a difference in how far things are able to travel between these two. The production of an action potential or a graded potential is going to depend upon the existence of a resting membrane potential, as well as the presence of various ion channels, which we'll talk about in just a minute. So a membrane potential is an electrical potential difference, also called a voltage difference, across the membrane. So in excitable cells, this voltage is going to be called the resting membrane potential. So this resting membrane potential is going to exist because of an electrochemical gradient between the inside and the outside of the cell there. Graded potentials and action potentials are going to occur because the membranes of that neuron are going to contain different kinds of ion channels that are either going to be open or closed in response to specific stimuli. So because the lipid bilayer of the plasma membrane is going to be a really great electrical insulator, the main path for currents to flow across that membrane is going to be through various different types of ion channels. So when ion channels are open, they allow for specific ions to move across the plasma membrane and down their electrochemical gradient. And just a brief little reminder, an electrochemical gradient is a concentration or a chemical difference plus an electrical difference. This is usually caused by disparages in ion concentrations. So as ions move, they're going to create a flow of electrical current that can change the membrane potential. So we have a couple of different types of ion channels that are present in neurons. The first one that we're going to discuss is the leak channel. So the gates of a leak channel are going to kind of randomly open or close. They're just going to alternate between open and closed randomly. Potassium channels, these potassium leak channels, are going to be more numerous than sodium channels. And potassium ion leak channels are going to be leakier than sodium ion leak channels. So because of this, the membrane's permeability to potassium is going to be higher than that of the permeability to sodium. These leak channels are going to be located on just about all cells, including the dendrites, the cell bodies, and the axons of all of the various types of neurons. Our next type of ion channel that's found in a neuron is the ligand-gated channel. These ligand-gated channels are going to respond to chemical stimuli. This is some sort of ligand binding to its receptor. So these are going to open or close in response to binding of a ligand in some sort of chemical stimuli. These chemical stimuli, these ligands, can be anything from neurotransmitters to hormones and even other ions. Ligand-gated ion channels are going to be located in the dendrites of some of the sensory neurons, for example, in pain receptors. And they're also going to be in the dendrites and cell bodies of interneurons and motor neurons. Our next type of ion channel that's found in neurons is the mechanically gated ion channels. These mechanically gated channels are going to respond to mechanical stimuli in the form of vibration, like a sound wave, or from changes in pressure or tissue stretching. And the force of this mechanical action is going to distort that channel so that it's moved from its resting position to a more open, to an open gate position. So examples of mechanically gated ion channels are going to be the auditory receptors in the ear, where that channel is being distorted due to the presence of sound waves, vibrations, and it's allowing the presence of those vibrations causes that channel to open, and ions are able to flow in and out. Other examples are going to include receptors that monitor the stretching of internal organs, as well as touch or pressure receptors in the skin. Our next ion channel is a voltage-gated ion channel. These are going to open in response to changes in the membrane potential or changes in voltage. It's these voltage-gated ion channels that are really going to be important in participating in the generation and the conduction of action potentials in the axons of all of the various types of ions. So when there's a change in the voltage, a lot of the times when it depolarizes, become less negative, this is going to result in the opening of that ion channel so that ions can flow in and out of that cell. And then this here is just a really handy table of the various types of ion channels that are found in neurons, as well as with a description of them, as well as the locations where they're typically found. So now we're going to talk a little bit more about the resting membrane potential of a cell, specifically a neuron. So the resting membrane potential is going to exist because of a small buildup of negative ions that are in the cytosol along the inside of the membrane. So you can see that here. We've got a little tiny buildup of negative ions right along that plasma membrane. And then an equal buildup of positive ions in the extracellular fluid along the outside surface of the membrane. Okay? So it's this just very slight difference here. We've got a buildup of negative on the inside, a buildup of positive on the outside. So this separation of positive and negative electrical charges is going to be a form of potential energy, which is measured in volts or millivolts. And this buildup here, this buildup of charge is going to occur only very close to the membrane. So the cytosol and the extracellular fluids elsewhere in the cell are going to contain an equal number of positive and negative charges and are therefore going to be electrically neutral. So it's this difference in charge here, this buildup of negative or positive charge, is only going to occur very close to the membrane. We can measure this resting membrane potential using something called a voltmeter. So the way this works is that the tip of a little recording microelectrode is going to be inserted inside the cell. And then a reference electrode, which is just a little device that conducts electrical charges, is going to be placed outside the cell in the extracellular fluid. The recording microelectrode and the reference electrode are then going to be connected to a little instrument called a voltmeter. And this is going to detect the electrical differences or the voltage across that plasma membrane. So in neurons, the resting membrane potential is usually going to range between -40 and -90 millivolts, with a typical value being -70 or -70 millivolts. So a cell that exhibits this membrane potential is also said to be polarized. All right? So if it has a -70 millivolt resting membrane potential, it's said to be polarized. So there are several factors that are going to contribute to this resting membrane potential. The membrane of the non-conducting neuron is going to be positive outside and negative inside, and this is determined by three different things. The first is going to be the unequal distribution of ions across the plasma membrane, as well as the selective permeability of the neuron's membrane to sodium and potassium ions. Our second factor that's going to contribute to this resting membrane potential is going to be the fact that most anions are unable to leave the cell. So most anions that are inside a cell cannot leave freely, unlike potassium. This is because they're typically attached to some sort of big, chunky, non-diffusible molecule, things like ATP, or some other large protein that can't just easily diffuse through the cell. So they're kind of trapped in there. And then our third factor contributing to the resting membrane potential is the sodium-potassium pumps, or a sodium-potassium ATPase. And this is going to expel 3 sodium ions for every 2 potassium ions that it imports. So since this pump is going to remove more positive charges from the cell than it's able to bring in, that pump is going to be said to be electrogenic. And this simply means that it's going to contribute to the negative resting membrane potential. All right, so we understand how that works then. So if something is removing more positive ions than it is able to bring in, it is making the membrane more negative, or the space by that membrane more negative. So it's electrogenic. So now that we've discussed the resting membrane potential, let's look at the differences in the role of the graded potentials and the action potentials in a neuron. So starting with the graded potential, this is a small deviation from a resting membrane potential that makes the membrane either more polarized, so inside is going to be more negative, or less polarized, where the inside is less negative, more positive. So when the response makes the membrane more polarized, i.e. the inside is more negative, this is termed a hyperpolarizing graded potential, right? So hyper meaning above, right? If you're hyperactive, you're overactive. So if it's hyperpolarizing, it's becoming more negative. If polarized means that it's got a baseline negative charge, remember negative 70 usually, millivolts. So if it's hyperpolarized graded potential, it's going to become more negative. It's going to go become, from minus 70 to minus 80, minus 90, etc. Right? That is a hyperpolarizing graded potential. When the response makes the membrane less polarized, i.e. the inside of the cell is less negative or more positive, same thing, this is going to be termed a depolarizing graded potential. So it's becoming less negative. It's moving up towards being positive, right? Depolarizing graded potential. A graded potential is going to occur when a stimulus causes these mechanically gated or ligand-gated channels to open or close in an excitable cell's plasma membrane. So typically, mechanically gated channels and ligand-gated channels are going to be present in the dendrites of sensory neurons, while ligand channels are going to be more numerous in the dendrites and cell bodies of interneurons and motor neurons. And these graded potentials are mainly going to be found in the dendrites and cell bodies of neurons. So let's look then at the stimuli, how stimulus strength is going to impact graded potentials. So to say that these electrical signals are graded just simply means that they are going to vary in amplitude or size, see here, depending on the strength of the stimuli. So the larger or smaller they're going to be large. These graded potentials are going to be larger or smaller depending on how many ligand-gated or mechanically gated channels are open or closed, and for how long each one is going to remain open. So if there are a lot of these ion channels that are open and they're open for a long time, that's going to cause a larger stimulus to occur, and there's going to be a greater difference in amplitude here because the strength of that stimuli is going to be greater than if there were, say, only a few ion channels open. Much smaller graded potential would occur. So the opening and closing of these ion channels is going to alter the flow of very specific ions across that plasma membrane, and this is going to produce a flow of current that is localized. This simply means that it is going to spread to adjacent regions along the plasma membrane in either direction from the stimulus source for a short distance, and then it's going to gradually die out as those charges are lost across that membrane through those leak channels. So this mode of travel by which a graded potential dies out as they spread along that plasma membrane are known as decremental conduction. So because they die out within a few millimeters of their point of origin, graded potentials are really only useful for short distance communication only. However, although an individual graded potential is going to undergo decremental conduction, it can also be made or become stronger and last longer by summating it with other graded potentials. So summation is the process by which graded potentials can be added together. If two depolarizing graded potentials are summated, the net result is going to be a larger depolarizing graded potential. And then if there are two hyperpolarizing graded potentials that are summated, the net result will be a larger hyperpolarizing graded potential. If two equal but opposite graded potentials summate, i.e. one that's depolarizing and one that's hyperpolarizing, they're usually going to cancel each other out, and the overall graded potential is going to disappear. So here you can see an example of summation of a depolarizing graded potential, where we have a stimulus and it's starting to die out with that decremental conduction, and then we add a second stimulus that would normally be the same amplitude as our first stimulus, but because they are summated, it makes it a larger overall, a more depolarized graded potential there. When it comes to naming graded potentials, they are going to have different names depending on which type of stimulus is going to cause them and where they occur. So when a graded potential occurs in the dendrite or cell body of a neuron in response to a neurotransmitter, it's called a postsynaptic potential. And the graded potential that occurs in sensory receptors is going to be termed a receptor potential. So now let's look at action potentials and how these are different from graded potentials. An action potential or a nervous impulse is going to be a sequence of rapidly occurring events that are going to decrease and reverse the membrane potential and are then eventually going to restore it to that resting state, that minus 70 millivolts. An action potential is going to have two main phases. There's a depolarizing phase and a repolarizing phase. During the depolarizing phase, which you can see here in green, the negative membrane potential is going to become less negative. It's going to reach zero, and then it's going to become positive. So it's depolarizing, it's becoming less negative, and then all the way into becoming positive. Then during the repolarizing phase, the membrane potential is going to be restored to its resting rate of minus 70 millivolts here. Following that repolarizing phase, there can sometimes be an after hyperpolarizing phase, and during this time, or during this phase, the membrane potential is temporarily going to become more negative than the resting membrane potential. So here you see our after hyperpolarizing phase. So let's look at why this happens. There are two main types of voltage-gated channels that open and then close during an action potential. These channels are going to be present mainly on the axon plasma membrane and on the axon terminals. So the first channel that opens is a voltage-gated sodium ion channel. When this is going to allow for sodium to rush into the cell and cause it to depolarize. Right? So our depolarizing phase, we've got the opening of our voltage-gated sodium channel, which is allowing for the influx of all of these positively charged sodium ions. Causes our resting membrane potential to go from minus 70 and become less negative because we're adding positive ions in there, positively charged ions, right? Sodium ions specifically, right? Becoming less positive until it is fully depolarized, right? This is caused by voltage-gated sodium channels. Then a voltage-gated potassium channel is going to open, and it's going to allow potassium to flow out of the cell, and this is going to produce the repolarizing phase. So positively charged potassium ions are flowing out of the cell, so the inside of the cell is becoming less positive, i.e. more negative, and we're having our repolarizing phase here. The after hyperpolarizing phase that can sometimes occur is going to happen when the voltage-gated potassium channels remain open after that hyperpolarizing phase is ended, and it's going to lead to a hyperpolarizing potential here, where it's more negative, right? So let's look at how stimulus strength impacts an action potential, or the formation of an action potential, and how that compares to a graded potential. So an action potential is going to occur in the membrane of an axon of a neuron when depolarization reaches a certain level, and this level is called threshold, right? So this is the level of which a neuron needs to be depolarized in order for an action potential to spark up, all right? So the generation of an action potential is going to depend on whether a particular stimuli is able to bring about a membrane potential, So the generation of that action potential is going to depend on whether a particular stimulus is able to bring that membrane potential to threshold, right? An action potential that doesn't occur, an action potential will not occur in response to a sub-threshold stimuli. This is just a stimulus that is too weak to cause a full depolarization to reach the threshold level. So a depolarization that cannot bring the membrane potential to threshold. Right? No action potential will occur here. However, if an action potential will occur in response to a threshold stimulus. Right? This is when the stimulus is just strong enough to depolarize the membrane to threshold. So it's depolarized enough that that action potential can take hold and run off. Several action potentials will be formed in response to something called a supra-threshold stimulus. And this is a stimulus that is strong enough to depolarize the membrane above threshold. So it's a really big stimuli. So each of the action potentials that's caused by a supra-threshold stimulus is going to have the same amplitude as an action potential caused by a threshold stimulus, but there's going to be a greater frequency. Right? So we're not going to have any difference in amplitude like we would see in our graded potentials, where a larger stimulus would result in a larger graded potential. With action potentials, a really big stimulus, a supra-threshold stimulus, is simply going to result in multiple action potentials of the same amplitude. But they're going to be the same amplitude as an action potential that is caused by just a normal threshold stimulus. Right? So that is a big difference between an action potential and a graded potential. Is that for an action potential, the size of the stimulus after it reaches threshold is going to cause an increased number or frequency of action potentials to occur, but they're all going to be in the same amplitude. Whereas a graded potential, the greater the stimuli, where it was with a graded potential, the larger the stimuli, the larger the amplitude for that graded potential. But it's going to just be the one graded potential still. So that's a big difference there between action potentials and graded potentials as far as the strength of stimuli that causes them is concerned. So let's look at the steps that are involved in creating these action potentials, specifically with regard to our sodium and potassium voltage-gated channels. All right, so we're going to start up here with this little picture in step 1 in the resting state. So during this state, all of the voltage-gated sodium and potassium channels are going to be closed. The axon plasma membrane is going to be at a resting membrane potential with a small buildup of negative charges along the inside surface of that membrane, and then an equal number of positive charges built up along the outside surface of the membrane. All right, so in our second step then, we're going to enter our depolarizing phase, which you can see here. During this phase, the membrane potential of an axon is going to reach threshold. The sodium channels are going to open, and as sodium ions move through these channels into the neuron, a buildup of positive charges is going to form along the inside surface of that membrane, and the membrane is going to become depolarized. So you can see that here in the graph. With the influx of positively charged sodium ions, our membrane potential is becoming less negative, reaching 0, and then becoming positive. It's depolarizing with the influx of these sodium ions. All right. After the depolarizing phase, we then reach the repolarizing phase, which you can see here. The sodium channel inactivation gate is going to close. So those sodium channels are closed, and the potassium channels are going to open. The membrane is going to start to become repolarized as soon as those potassium ions leave the neuron, and a few negative charges are going to begin to build up along the inside surface of the membrane. So this is when we get the beginning of our repolarizing phase. We've got this flow of positively charged potassium ions out of the cell, which is making that membrane potential become more negative. It's going back down, trying to get to negative 70 millivolts. Right? As this repolarizing phase continues, potassium is going to continue to flow outward, and as more of those potassium ions leave the neuron, more negative charges are going to build up along the inside surface of the membrane. So this potassium outflow is eventually going to restore the resting membrane potential, and sodium channel activation gates are going to close, and the inactivated and inactivation gates are going to open. So this is going to return the resting return. This is going to cause the return to a resting state when the potassium gates close. Right. So you can see here we've got our outflow of potassium ions, which is bringing us back down to our repolarized, our minus 70 millivolts. And then the whole process, the whole cycle can start again. So let's talk about something called refractory period, just real quick. That's specific to action potentials. So the period of time after an action potential begins, during which an excitable cell cannot generate another action potential in response to a normal threshold stimulus, is going to be called a refractory period. So during the absolute refractory period, even a very strong stimuli is not going to be able to initiate a second action potential. So no matter what you do during that refractory period, you're not going to be able to spark up another action potential. This period of absolute refractory is going to coincide with the time during which the sodium ion channel activation and inactivation is occurring, and then inactivation of the inactivated sodium ion channels are not going to be able to reopen. So they must first return to that resting state before they can reopen, and you can spark up another action potential. So no matter how much stimuli you pump into that cell, there is not going to be another action potential that happens during the absolute refractory period. On the other hand, the relative refractory period is going to be the time during which a second action potential can be initiated, but only by a really big, larger than normal stimulus. So it's going to take a little extra oomph to get that action potential to spark during the relative refractory period. You can still get it, but it takes a lot more effort than during normal depolarization. So this relative refractory period is going to coincide with the period when the voltage-gated potassium channels are still open after inactivated sodium channels have returned to their resting state. So in contrast to an action potential, which has this refractory period, graded potentials do not exhibit a refractory period. So that is another big difference between graded and action potentials. So just kind of a fun fact, local anesthetics, things like lidocaine or procaine, as well as some neurotoxins like tetrodotoxin from the pufferfish, are going to insert themselves into the voltage-gated sodium channels, and this prevents them from those sodium channels from opening. So that nerve impulse is not going to be able to pass that obstructed region. So because of this, the action potential is not going to be able to propagate past that obstructed region. So the pain signals from whatever part of the body that you've just applied our topical or local anesthetic are not going to be able to reach the central nervous system where they're going to be consciously perceived. So that's how those work. They work by simply blocking those voltage-gated sodium channels. So here is a very handy little table, just looking at a comparison of the graded potentials and action potentials that are found in neurons. So let's look now at the propagation of these action potentials. So in order for communication to occur from one body part to another, action potentials must travel from where they arise at the trigger zone to the axon terminals. Action potentials are not going to die out. They're not decremental like what we would see in a graded potential. They are going to keep their strength as they spread across the membrane of a neuron. And this spread across that membrane of the neuron is called propagation. So there are two main types of conduction that can happen for these neurons. Right? The first is called continuous conduction. This is going to involve a step-by-step depolarization and repolarization of each individual segment of the plasma membrane. Right? So in continuous conductions, ions are going to flow through their voltage-gated channels at each adjacent segment of the membrane. And it's important to note that the action potential propagates only a relatively short distance in a few milliseconds. So continuous conduction is going to occur in unmyelinated axons and in muscle fibers. So here you can see we've got our depolarization, repolarization of this one little region of our plasma membrane, which is going to cause the depolarization, repolarization of our plasma membrane and the little region next to it on and on and on as it propagates down that axon, right? This is only going to occur in unmyelinated axons, and it's a little bit slower. Our second type of conduction is called saltatory conduction, and saltatory just means leaping, and you'll see why in just a minute. This is a special mode of action potential propagation that occurs along myelinated axons, and it occurs because of the uneven distribution of voltage-gated channels. So when an action potential propagates along a myelinated axon, an electrical current that's carried by electrons is going to flow through the extracellular fluid that surrounds our little myelin sheath. You can see it flowing around that myelin sheath, and it's going to flow through the cytosol from one node of Ranvier to the next, right? So it's kind of leapfrogging over that myelinated bit to get to the node of Ranvier. So the action potential at the first node is going to generate ionic currents in the cytosol and the extracellular fluid that is going to depolarize the membrane to threshold. And this is going to open the voltage-gated sodium channels at the second node. All right. The result is going to be ionic flow through the opened channels that's going to constitute an action potential at the second node. Then the action potential at the second node is going to generate ionic currents that are going to open the voltage-gated sodium channels at the third node, and so on and so on. So it's kind of leaping from one node of Ranvier to the next. So each node then is going to be repolarized after it depolarizes. So it's going to do the normal cycle of depolarization and repolarization, but it's going to cause that electrical current to kind of leap from one node of Ranvier to the next. So nerve impulse conduction, in which the impulse jumps from neurofibril node to node, is called saltatory conduction. That's why we call it that. Saltatory meaning leaping. That's how it got its name. So the flow of current across the membrane only at the nodes of Ranvier is going to have two main consequences. The first is that the action potentials are going to appear to leap from node to node as each nodal area is depolarized to threshold. So because an action potential leaps across long segments of that myelinated axon, as current flows from one node to the next, it's going to travel much faster than it would in an unmyelinated axon of the same diameter. The second consequence is that there's going to be a smaller number of channels that need to be opened at one time. They're only going to need to open at those nodes, rather than many channels in each segment of the membrane having to open. This is going to be a more energy efficient mode of conduction. So because only those small regions of the membrane are going to depolarize and repolarize, there's going to be minimal inflow of sodium ions and a minimal outflow of potassium ions. It's going to occur each time an action potential passes by. Therefore, less ATP is going to need to be used by that sodium-potassium pump to maintain the low intracellular concentration of sodium and a low extracellular concentration of potassium. So it's more energy efficient. Less ATP is needed to maintain the proper levels of sodium and potassium within the cell. There are going to be several factors that are going to affect the speed at which propagation can occur. So the propagation speed of a nerve impulse is not going to be related to the stimulus strength, like what we would see in a graded potential. However, the axon diameter is going to impact propagation speed. So a larger diameter fiber is going to conduct impulses faster than those with smaller diameters. We have three different types of fibers that are going to make up axons. The first is going to be an A fiber, and this is the largest diameter axon, and these are myelinated. The second is a B fiber. These are axons that are about 2 to 3 millimeters in diameter, and they can be either myelinated or unmyelinated. And then our smallest diameter axon are the C fibers, and these are all unmyelinated. So this diameter of the fibers that make up that axon is going to impact how fast an action potential can propagate. So the action potential is going to be able to propagate fastest through our A fibers with their really large diameter. The second factor that is going to impact propagation speed is myelination. So myelinated fibers are going to conduct impulses faster than unmyelinated fibers, like what we talked about with the continuous versus the saltatory conduction. And then last but not least is temperature. Nerve fibers are going to conduct impulses faster when they're warmed versus when they're cool. So those are the three things that are going to impact the propagation speed of an action potential. And that is all we have for this part of the lecture.